Good morning, guys. I am Dr. Shashikala, Associate Professor, Department of Zoology, Maharani Lakshmi Ammani College. Today we will discuss about the gastrulation. So this is a part of the developmental biology in paper 6. So first coming to the definition of the gastrulation. What is gastrulation? So in last class we have discussed about the blastulation. Okay, blastulation in sea urchin, frog and chick. Today we will see about the gastrulation. Gastrulation is the process of second stage of development. Okay, gastrula is the second stage of development. Gastrulation is the process of forming the gastrula. Okay, so this is early development of any organism, so that is called as the embryonic development. So during the embryonic development, the cells after the blastula, the cells will undergo morphogenetic movement and it will form the three germ layers. Okay, so that is the ectoderm, endoderm and the mesoderm. The formation of three germ layer is called as the gastrulation. Okay, so gastrula is a second stage, as I said. Okay, so in some organism, the gastrula may be a diploblastic. So in the sense, it may have the only two layers. In some, it may be a triploblastic. Because you know that some of the organisms will have only a two layers, diploblastic organisms. Some will have the three layers or the three germ layers, ectoderm, endoderm and the mesoderm. This is the triploblastic. Coming to the definition of gastrulation is the process of formation of gastrula. Okay, so and it is the gastrulation is the most prominent morphogenetic event in the early development of any organisms. Okay, so results of the gastrulation, there is a formation of gastrula. So coming to the definition of gastrula, there is a difference between gastrulation and gastrula. The embryonic stage next to blastula. The cells of blastula move in orderly fashion so that they take up new position in gastrula to form three germ layer, ectoderm, mesoderm and endoderm, and with new cavity called archenteron. So archenteron will open exterior through the blastopore and this stage is called as gastrula. Is it clear? Gastrula and the gastrulation. You can see the picture here. So that is the first one is the blastula. In the blastula you can see that only a single layer of cells with the large cavity at the center. In the gastrula you can see that all the three germ layers, ectoderm, endoderm and the mesoderm with centrally located cavity called archenteron. Archenteron will be a opening outside through the blastopore. Next, coming to gastrulation, the morphogenetic movement. So there is six basic movements of the cells. Okay, so that is called as the formative cell movements. These six basic movements are common for all the organisms during the gastrulation. Okay, so any organisms will undergo these six formative cell movements. Okay, so the basic types of formative cell movements are invagination, it is also called as emboli, involution, epiboli, ingression, delamination and convergence and divergence of the cells. First you can see in the picture the invagination. So this is also called as the tucking in, otherwise the infolding of the cell sheet into the embryo. So this is called as the invagination. Okay, so here invagination will take place from the vegetal pole, otherwise from the lateral side of the blastula. Okay, the cells will move inside. Okay, so that is a process of invagination involves the tucking in of cells. Okay, so this makes an indentation on the surface of the blastula. So therefore this appears like a notch. The cells move in without disturbing entire layer of cells. Here you can see that. Next, coming to the involution. So involution is also called as rolling in due to the process of epiboli. Epiboli is nothing but the overgrowth. Epiboli, the cells near the blastopore get pushed and they have to roll inwards and move along the inner surface of the outer layer of the cells. Here you can see in the second picture. So this is the blastopore. Due to the epiboli, so there is a overgrowth. Okay, so overgrowth of the cells and all the cells will come and accumulate near the blastopore and these cells will slowly move inside. Okay, so that is called as a rolling in, otherwise the involution. Here you can see that this is the outer layer of the cells. The cells will move the inner surface of the outer layer of the cells. Next, coming to the epiboly. As I said already, epiboly is nothing but the overgrowth. Okay, so here towards the animal pole there is a smaller cells, otherwise the micromeres. Okay, so here towards the animal pole there is a micromeres. Okay, so what will happen? The micromeres immediately they will undergo division and they will overgrow on the macromeres. Okay, so completely it will covers the embryo, otherwise the blastula. So except near the blastopore. Okay, rapidly divides and it will grow over the larger vegetal pole cells. Those cells are the future endodermal cells. Okay, so this is the next step. Next, coming to the convergency and divergency of the cells. The cells that grow downwards all move towards the blastopore, and this process is called as the convergency. Okay, so the cells will go and converge near the blastopore. So from the blastopore the cells will involute, otherwise they will roll in, and from there it will move to the different parts of the embryo. Okay, so that is called as the divergency. Okay, diverge is nothing but the move upwards, and they will occupy the either side of the notochordal cells and also the endodermal cells at the dorsal side of the early gastrula. Okay, next coming to the ingression. Ingression is the next type of morphogenic movement of cells. So here individual cells they will move from the layer into the cavity. Okay, so the cells will move from the layer, they will move into the cavity. The cavity is nothing but the blastocele. Okay, so there is a blastocele. So the cells will move to the blastocele. So that will move into the cells. So this is called as the ingression. Individual cells will move. Okay, so next coming to the delamination. So this is the process where the single layer or sheet of cells is split into forming two layers. Okay, so this is called as the delamination. So there is a two parallel layers will form. Next, we will move to the gastrulation in sea urchin. So, blastulation in sea urchin we have discussed already in the last class. Next, we will see the gastrulation in sea urchin. So here gastrulation in sea urchin, so this has been broadly divided into two distinct phases. There is a primary invagination and the secondary invagination. So both the primary invagination and the secondary invagination has been explained in 4 steps. Okay, first, second and the third and the fourth step. So this division is for the conveniency of the study. Okay, so we will see the first step. Here you can see in the picture. Okay, so this is the first step. In the first step, the presiding the occurrence of the invagination. So the invagination will take place at the vegetal pole. Okay, the cells around the vegetal pole, it will become elongated and they will give rise to thickened vegetal plate. Here you can see that in this part it will give the thickened vegetal plate. Okay, so now what will happen? The ingression of the primary mesenchyma cells will take place. So the cells which are present near the vegetal pole, so this is a single layer of cells. Some of the cells, they will lose their cilia and also they will lose their adhesion with the neighboring cells, and just they will move into the cavity, that is the blastocele. Okay, so these cells are called as the primary mesenchyma cells, otherwise the PMCs. Okay, primary mesenchyma cells. Now, these primary mesenchyma cells, they will begin to migrate into the blastocele. They will begin to migrate into the blastocele. Here you can see that. So, this is the primary mesenchyma cells. So, this is the first step. So here the ingression of primary mesenchyma cells, it is accompanied by the retraction of, as I said, the anchoring microvilli from the hyaline layer. They will lose their microvilli and they will detach from the hyaline layer, and also they will lose the adhesion between the neighboring cells. So there is a disruption of the basal lamina that coats the inner surface of the PMCs. So here retraction of the cilia and destruction of the microtubules, because inside the cell the microtubules which will help to hold the cells in columnar shape. Okay, so here due to the destruction of microtubules, this columnar cells will become round in shape. Okay, so now PMCs are round in shape and they will slip from the vegetal plate into the blastocele, and there will be a migration and the patterning of the PMCs in the blastocele. You can see in the picture, you can see here, these are the primary mesenchyma cells. Okay, so they will become round in shape. Initially they were columnar cells, now they will become round in shape. Next, coming to the second step. So second step, it is a primary invagination. So here what will happen, unlike in amphibians. Okay, so here amphibians, you know that there is a apical constriction of the bottle cells. So it will lead to the formation of the dorsal lip. So here in sea urchin, these bottle cells are arranged in circle at the central region of the vegetal plate. The bottle cells are arranged in a circle at the central region of the, yeah, here you can see that. See, these are the bottle cells. Okay, so these are arranged in a circle at the central region of the vegetal pole. Now they will fold inward, otherwise imagination will takes place. See here, so this is the forming the small dent. So this is a blastopore. Okay, so here the some cells will change their shape. Those are nothing but the primary mesenchyma cells. And next what will happen, the bottle cells which are present near the vegetal pole, they will arrange themselves in the form of circle and slowly they will be invaginating into the blastocele. Okay, so this is the primary invagination. So here the thickened vegetal plate, no, it will bend inversely and it will form the short stub-like gut rudiment. So this is the primary invagination. So here the bottle cells are, it is very clear that bottle cells will trigger the initially inward bending of the vegetal plate. Okay, so due to the bottle cells, there is a initial, otherwise the primary invagination will takes place. Next, coming to the step 3, so there is a elongation of archenteron will takes place. So what will happen during the couple of hours after primary invagination, the gut rudiment, no, so this elongates, gut rudiment, so which was like a short stub like, so this will become elongates. Okay, so here when it will elongates, the another population of the cells, so that is called as the secondary mesenchyma cells. Okay, another population of cells that is called as the secondary mesenchyma cells. Here you can see in the picture, this is the mesenchyma cells. So those cells move to the tip of the rudiment gut folds. Otherwise this is the invaginated, okay, rudiment gut folds. So here the secondary mesenchyma cells, otherwise these are also called as the SMCs. They will move to the tip of the gut rudiments. So now the SMCs, so they will extend a thin and long phylopodia towards the animal pole. Okay, so this SMCs, they will extend a thin and long phylopodia towards the animal pole. Okay, so this will explore the attachment towards the animal pole. Here you can see that this is the phylopodia. This is very smaller one, very thin. So it is around 0.2 to it is 0.4 micron in diameter, and it will be extending from the SMCs. That is the nothing but the secondary mesenchyma cells. Now these thin phylopodia which will provide the contact with the several cells. Okay, so several cell diameters away, because this phylopodia is very long, thin, flagella like. So the cells are contact with the animal pole cells. Now the gut rudiment of the vegetal plate. So here gut rudiment of the vegetal plate, it will be pulled because this phylopodia will be attached to the animal pole cells. Now you can see that the elongation of the archenteron will takes place. The small rudiment, gut rudiment will become elongated. Here you can see in the third picture, it will elongate and also it will opens exterior through the blastopore. So when it will elongates, the phylopodia, it will become shorter. Okay, phylopodia will become shorter and shorter. Finally, the phylopodia will disappear and here secondary mesenchyma cells will have adhesion with the animal pole cells. Okay, so this will have the adhesion with the animal pole cell. So that is the fourth step. So this is called as the secondary invagination. So where is the slender archenteron will become the elongated, the morphogenetic movement is called the secondary invagination. After such a phase of archenteron elongation, the gut rudiment rapidly it will elongate until its tips reaches, as I said, inner surface of the apical plate, that is the secondary invagination. Now the archenteron elongates and the tip of the gut rudiment will reaches the animal pole. That area is called as the apical plate. Okay, so this is called as the apical plate. So this area will become presumptive mouth. So okay, so archenteron where it will meet the ectodermal tissue, so that will become the presumptive mouth. Means future this part will become the mouth of the larva. So anus will form blastopore. The opposite, the blastopore will form the anus. So here it is also been thought that the contraction of the phylopodia, the contraction of the phylopodia will help to connect the archenteron tip and the apical plate. Okay, so plus the gut rudiment upward. So now the single layer has become double layer and also in between the mesodermal cells will arrange. Okay, so now the gastrula is the three layer, that is the ectoderm, endoderm and the mesoderm, and also it has got the central cavity, that is the archenteron. Okay, so in recent years, it has been also elucidated that the recruitment of the presumptive endodermal cells, so this will last to the mid or late prism stage. Okay, so that is the prism stage of the sea urchin. Okay, so this is about the gastrulation in sea urchin. The gastrula of sea urchin, if we'll see, it will have the three layers like ectoderm, endoderm and the mesoderm with centrally located cavity called archenteron. Okay, so then it will changes into a pluteus larva. So here in this diagram, you can see the process of gastrulation. Okay, today we will stop here. Next class we will discuss about the gastrulation in frog. Thank you guys.